Welcome back friends, welcome to another lecture from Just Biology. In this lecture we're going to talk about elongation process in prokaryotic cells. In the last lecture we have talked about initiation process of prokaryotic DNA replication. In this lecture we're going to talk about elongation process. So we know that the elongation process, the word, that word means elongation, just the lengthening of the DNA strand. So that is the idea. The elongation means just polymerization of the strands of the DNA. We can call it as elongation process. We can see how this process is catalyzing by the prokaryotic cells. What are the things they need of fulfilling elongation process in prokaryotes? So we know that they will form a replication fork. So we will form a replication fork for the replication to begin. Just beginning of the elongation process, they made all the fork and they assembled with many enzymes and proteins as well. Now, so I told you the directionality of the DNA replication. So they usually run with 5' to 3' directionality they followed. So here 3', here the 5', and here the 3', and here the 5'. Now this is the hydrogen bonds, the broken hydrogen bonds. This is also, here also you can see the many bonds. Now I have told you the rejoining of these two strands are prevented by a specific protein that is called SSBP. So we can call it as SSBP, single strand binding protein. So we can call it a single strand binding protein. The SSBP, will the many copies of SSBP you can see over the strands, because they pretended to form a double strand of the DNA. So they will not allow the strand to rejoin. So that is the function of SSBP. Now, so they made all these setups. Sorry, I forgot to mention this complex. So this is the complex I said before. Yes, no. This I have mentioned earlier. So this is the complex. So BNC complex. Now this is the scenario. Now let's move on to the next level of the bacterial replication. If you take any biological process, they need a starting material. They need some precursor molecule. But in case of this prokaryotic DNA replication, they need RNA nucleotide. So this is RNT. That is ribonucleotide for starting the process. So we can call this RNA nucleotide as a primer. So we can call it as primer. So this is the primer. Sorry. So but I should select some other color, so better for the better visibility. So this can be a prime. This is 5', and here you can see 3' OH. So this is the primer which is bound with, or which is attached with the strands, one of the strand. Now, so this I have to mention here. So this is RNA primer. And the question is, why they need RNA primer? So that is the question here. So because we cannot replicate, they cannot replicate DNA with DNA primer. No, they cannot start the function with DNA primer, because the DNA primer which will not provide 3'OH group or 3' hydroxyl group. It should provide 3' OH group. That's why the system itself select the RNA primer. Now the question is, who will give the RNA primer, or how the RNA primer will be formed during this stage? You can see there is a protein which synthesizes RNA primer inside the cell. The protein which helps to synthesize RNA primer is DNA G protein. So this is the protein which synthesizes a primer. So DNA G protein, we can call it as DNA primer, or the primase. So we can call this DNA G protein as a primase enzyme because this will synthesize this RNA primer. Just look at here. So this DNA G protein will synthesize RNA primer. But in case of eukaryotes, you can see the same DNA G protein but different name. So in eukaryotes, we can call this G protein as primase. So we can call it as a primase. So this is the prokaryotic primase, and that if the G protein which is present in the eukaryotes, we can call it as primase. So this is prokaryotes G protein and eukaryotes primase. So this is the scenario. Now they have got the ribonucleotide and all. Then what which enzyme supposed to be comes in action? So we need to polymerize this strand. Now the enzyme which come forward for the synthesizing or the polymerization is DNA polymerase 3. DNA polymerase 3. Actually, this DNA polymerase 3 will have only the polymerizing activity. So they will add this nucleotide with respect to the RNA primer. So wherever they find the primer, they started to polymerizing the nucleotide with complementary nature. So if the adenine is present here, it's supposed to add thymine. If it is G, it's supposed to add a C. So this is the action of DNA polymerase. Now just remember the structure of the DNA polymerase. Just remember this. This is thumb, palm and finger. This is the structure of, it's resembled to that of this hand like structure, or the thumb, palm, finger. So this is, so here we can say this is the catalytic center or the catalytic region of the polymerase. Here the catalysis will happen, but this portion, this thumb will take the nucleotide, will bring the nucleotide and just fix it over here. Then the fixing, whether they are checking the correct base or not, or they are checking, the proofreading will be done by the finger like things in the DNA polymerase. So actually they act as a finger, palm and the thumb. So this is the structure of DNA polymerase. Now let's say, so DNA polymerase can move, just remember the directionality. So I have mentioned in the last lecture, they always follow a specific directionality. So they will form like 5' to 3' direction. The DNA polymerase always drive through the 5' to 3' direction. So that is the directionality of the DNA polymerase. Let's imagine this in this strand. So we can call it as strand A. This is B. So let us understand better way. We can call this strand, the 5' to 3' can be A, and the 3' to 5' can be B. Now the A, if you see the A strand, the directionality of this polymerization is, it's quite good, because so they follow the rules. So 5' to 3'. But if you see here, so here is the problem comes. But the directionality, actually the directionality of this B strand is supposed to be 5' to 3'. So here. So the direction will be in this direction. So here the problem comes. The DNA polymerase cannot act simultaneously at the different direction. So in this direction DNA polymerase is quite fine. But they cannot synthesize, they cannot synthesize the new strands over here. You know why? Because they are a complex enzyme, though they are complex in nature, or they are they are well efficient nature, but they cannot drive, they cannot move it parallel as well as the anti-parallel direction simultaneously. But in such case, so what the DNA does, so they will make or they will rearrange themselves. And this is the important part of a DNA replication, the rearrangement of the strand. So this is the, actually if I want to talk about this rearrangement in DNA, I'm not going to talk about all those things much on detail. Just remember how this, how this DNA and when they are rearranged. So we'll discuss that alone, because that is more than enough for us to understand the elongation process. Now just see, you just see here. So what the DNA does here is, actually this, this scenario, so based on this directionality, we can call this DNA in, different names, because this is continuously synthesized strand. You can see they synthesis continuously, without any interruption or without any disturbance. They can synthesize, they are able to synthesize in this direction. But in this direction, if you see, they have to stop at somewhere else, and again they have to start. So they have to stop somewhere else and again they have to start. So this is discontinuous strand. So based on this issue, we can call this first strand, strand A, that is, we can call it as leading strand. So we can call it as the leading strand. Why? Because they are not interrupted. So they are a continuous strand. So they can synthesize continuously. So there is no interruption without any problem. But if we see this B strand, so they are discontinuous. They will stop somewhere else and they will start from where did they stop. So that we can call it as lagging strand. So we can call it as lagging, and that is leading. So these are the two names, we call them in a particular, stage, or that is elongation stage. Now, as I told you, so they will possess some kind of rearrangement. How they rearrange? So that is, I'm going to tell you now. So let's erase this strand. I have to draw it again for the explanation. So we'll see. So here you just see, so this is the lagging strand here. What actually usually they do for the synthesis of the new strand, so we have they will turn one of these strand. So they will turn one of these strand for the synthesis of new strand. So this is the 5' end direction, so the 5' to 3' direction. Here also they will turn the DNA for the 5' to 3' direction. So that is the idea here. Now just imagine this DNA is turning up. So this is turning, just make it turn for the synthesis of lagging strand. So this we should understand clearly here, because so this is a little bit confused. You may confused in this region. So that's why I'm telling again and again. So this this DNA strand will be turned. Just make it turn for the equalizing the two directionality, because the enzyme cannot drive through this direction. One is 5' to 3', this is 3' to 5'. So they cannot drive along with the DNA polymerase, so that they will make it turn. So how? So just they will make a turn. Sometimes there are many concepts in this turning process. They are just make a loop. This is a loop formation actually. So there are some loop like butterfly loops and some other kind of loops. So that I'm not going to discuss much on that. So just remember, this is the scenario. So they will make it turn. They will make it turn, and this strand is supposed to be, just make it turn like this. So just make it turn like this, so that this enzyme can read this direction very easily, like this direction, this A strand. So so in this direction, let's say I have to erase some of the portion here, so it's better to erase here. So I have to, so let's say this is a lagging strand, and this is just turning up in this direction. And here the DNA polymerase, so that is a large complex of DNA polymerase can polymerize both of these strands together. So this is the idea. So they will they will synthesize a new strand here. So this is the new strand and they will start to synthesizing in this direction, and here also, so they will synthesize in this direction. And just remember one thing, so as I told you, so they will not have any continuous synthesis. They have some interruption during the synthesis. Here you can see a multiple primer will be added here. So this is 5 prime to here the 3 prime. So here also 5. So here they will add some multiple primers for the synthesis. So this is the primer, the black which represents the primer. Here also they will have primer. Here also some different parts of the lagging strand would be attached with the different primers. This primer is supposed to be removed once the strand is synthesized. Once this process been over, we have to remove this primer. Then who will remove this primer? So this is obviously RNA. So we need some protein which degrade or which remove the RNA primer. So one such protein we can call it as RNase H. RNase H. This RNase H will remove this primer, or this primer will be removed by RNase H, and instead of this RNA primer, so we need to add nucleotides. We have to fill this gap actually. For the filling of this gap, the system will recruit another enzyme or another protein called as DNA polymerase. DNA polymerase 1. So this is DNA polymerase 1. What it does? DNA polymerase 1 will fill the gaps. So wherever they find the gaps, so they will fill with appropriate nucleotide, and the gap is sealed, or the gap will be filled by DNA polymerase. Then if you see this primer region, so of course they will fill the gaps. DNA polymerase will fill the gap, of course, but they cannot form that bond, the bond between this gap. So here let's say, imagine, so here they are synthesizing nucleotide. Let's imagine, so here they are synthesizing nucleotide, but this is sealed actually, so sealed by DNA polymerase, and this region, so you can see a gap. So that gap, that means we can call, actually that is not a gap. So there is a difference between gap and the bone, the region where not formed any kind of bond. This region we can call it as nick. So we can call it as nick. Nick means there is a base. Actually the base would be present. The different kinds of bases would be present, but the bond formation between two bases is absent. So that is the gap. That's why we can see a small gap between two bases. Such gap we can call it as nick. So we need to seal that nick. The nick will be sealed by a kind of enzyme. It's called as ligase. The ligase is a complex enzyme, so which fill the gap or which fill the nick. So nick will be sealed by ligase enzyme. The ligase will induce the formation of phosphodiester bond. So this is the scenario of the elongation of the prokaryotic cells. Actually, the one more thing I have to mention here, I think I have to discuss the bond formation between two bases actually. So let's as the enzyme is moving, as the enzyme is polymerizing the bases, how this bases is binding together, and which base, what is the chemistry between the polymerization. So I'm going to talk about a chemistry between the polymerization. So you can see, so this is the template strand. So you can take, this is template strand, and this is a newly synthesized strand. And if you see here, so they will have a certain 3' OH. So 3' OH group. You can see, just this basis, and the upcoming bases which is gonna bind with this complementary region. You can see a phosphate, 3-phosphate, because they are triphosphate form. So any bases. So this is one of the bases. So that doesn't mean thymine, because I'm just representing the base. So this is a scenario of the bond formation. Now let's look at it. So we can call this as alpha phosphate, beta phosphate, and gamma phosphate. So we can call these 3 phosphate into different names: alpha, beta, and gamma. Now, so this is a 3-OH. So we know that they have a lone pair of electron. They have a lone pair of electron. So that lone pair of electron will have a certain property. We can call it as a nucleophilic attack. So they will attack alpha phosphate. So they will attack alpha phosphate by nucleophilic attack. So as the result of this nucleophilic attack, these 2 phosphate, that is beta and gamma phosphate, will be released out. Just remember, 3-OH of this base will have a lone pair of electron. So this lone pair electron will attack alpha phosphate by nucleophilic attack. Then as the result of this nucleophilic attack, the beta and gamma phosphate will be released as a pyrophosphate, PPI. So so as the results, once they are removed from the bases, so they start to forming a bond. We can call it as phosphodiester bond, because the 2 phosphate will be removed from the structure. So that's why we can call it as phosphodiester bond here. So they form a bond over here. Then what is the fate of this PPI? So this PPI, they have the ample of a plenty of energy in it. If in human body we don't need any kind of external source of energy, because we have a lots of PPI. PPI will serve as an energy source in cells. So that's why we can call it as this cells, this kind of process is thermodynamic, because they don't need any energy, or they do not depends on the external source of energy. Bond formation is very important, because as so they are forming a bond like this, because they hit on the alpha phosphate, and this alpha phosphate will remove, that will kick out beta and gamma phosphate, and the beta and gamma phosphate will form an energy source. So this energy source will catalyze all this function, and as the result we got the phosphodiester bond. So this is the idea of the elongation process. I hope this video will helpful to understand the concept of elongation, and of course if you like the video, please hit the like button and don't forget to subscribe my channel.